So next up, we have Eric Wada, who also took long trips with me. He'll probably recount a few. One that he won't recount is when I asked him to stop and help me out with this snake that was in the middle of the road. So we're in the middle of somewhere in Texas, and there in the middle of the road was at least a 6-foot-long rattlesnake with a really distended gut. So it obviously just eaten something. It wasn't going to move or attack us. I said, Eric, go over there and poke it so I can get a nice picture. And he goes, Eric, fresh out of San Francisco, said, absolutely not.
Thanks, Andy. I actually have a picture of that snake, I think, but I did not put it in the presentation. Thank you, Andy, for welcoming me here today. I'm honored to honor you. A little bit about me. I'm Eric Wada. Worked with Andy from 2000 to 2008, so it was a long time. Typically, I was doing a lot of teaching. I taught introductory biology, Biosci 1C here when it was still the 1 sequence, as a TA for many quarters, and then also during the summer sessions. I also moonlighted teaching in the evenings at a community college, American River College here in the area, and that sort of inspired me to pursue teaching as a career at the community college level. So I find myself now in my 15th year as a full-time faculty member at Folsom Lake College. It's a 2-year community college, also in the Los Rios Community College District in the area. I want to point out that we do have a budding viticulture and enology program up in our Placerville Center, so I wanted to mention that. And also someone who might be familiar to Andy's lab and some of his members, Cecilia Osorio, is teaching in some of our viticulture courses as well, so some of you might recognize her name. In addition to teaching biology, I find myself doing a lot of committee work. Over the past few years, I've chaired my college's curriculum committee. I'm finishing my first year as the academic senate president of my college, and I've also stepped up to statewide service, serving next year as a North representative for the academic senate for California community colleges. What keeps me grounded, though, is the teaching. I very much enjoy the teaching still. How I got into the Walker Lab is I was a first-year graduate student in the plant biology graduate group during rotations. I was originally interested in disease resistance and systematics, two very different fields, some folks might say. Andy's research profile seemed to match really well with what I was interested in because of the disease resistance, and also his bioprofile suggested that he was interested in systematics of Vitaceae. It seemed to also complement some work that I was beginning in Dan Potter's lab, so I did my PhD in collaboration with both Andy and Dan. Andy had a lot of ideas regarding questions related to systematics. The first several months were dedicated to the questions at the family level, systematics of Vitaceae, with a focus on this plant here, Muscadina rotundifolia. I remember the first time I got to try a muscadine on one of our many trips to the vineyards on the west side of campus. And for anyone who has ever tried a muscadine for the first time, you probably don't forget that experience. The initial challenges in doing this work were mostly technical in nature, struggling with techniques to get quality DNA from molecular studies. After that, we quickly learned that the regions that we were working with were high in guanine and cytosine, so it made it difficult to get PCR reactions amplified and then also to sequence those materials as well. Meanwhile, I was also writing grants. I was still an early graduate student. Many of those were not funded. So I had a lot of problems, experienced a lot of challenges in the first couple of years of grad school. And I remember Andy's door was always open. It was really great to be able to consult with him. Gene mentioned earlier, he was always busy, always working, always the first one in the lab or in the office. Yet he always made time for his students, and I think that just that was really helpful in me being persistent in grad school, so I really appreciated that. His subtle guidance, his pep talks, those are what I think kept me going. Andy's research in breeding for Pierce's disease resistance was also progressing at that time, and that's when we also started shifting our interest from questions related to the whole family to more focused questions on the genus Vitis. And initially we were interested in the species of the southeastern United States, where Muscadina is found, but eventually we then shifted later to southwestern and the western species. Andy mentioned we went on a lot of collecting trips. So research in Andy's lab means going on a lot of collecting trips, and of these, the first trip in 2003 is perhaps amongst the most memorable, as it involved the greatest amount of driving while yielding the fewest number of specimens. This involved a red-eye flight to Dallas, which is like only 2.5 or 3 hours. If you get a good tailwind, it's probably 2.5 hours, and so you don't really sleep on that flight. And a connection to San Antonio. Bleary-eyed, we got in a car, and Andy never likes checking bags. And so this means going to a sporting goods store or a Walmart or something like that to pick up your cooler and your collection materials. I sort of had this trip kind of all planned out beforehand. I thought, okay, first thing we need to do is if we're going to be out there for a long time, we need to keep our plants kind of fresh, or at the very least, we need to pack some desiccant. And I remember we spent a lot of time trying to figure out how can we get some desiccant, a large enough amount of it, put it in my carry-on bag, goes to security, and security takes it away. And Andy says, it's your dry humor that's caused this. And I thought, okay, this is evidence of Andy's sharp wit and quick humor, making light of the situation at midnight, right before a mid-con red-eye flight. Anyway, after that trip, I learned the hard way that it's important to do research up front on where grapes can be found. So at this time, digital herbarium records were in their infancy. So it involved looking at those and also contacting herbarium directors to get an idea of where the grape populations were found. It was also after 2003 that I invested in a digital camera. So earlier collections involved those trips to Texas and Arizona, as I mentioned before. It was our trip to, so I guess I should share a few pictures here. These are not all sequential, but some pictures from Texas. The picture there in the upper right corner, welcome to Texas, drive friendly, the Texas way. You'll notice that the star there is it doesn't have bullet holes in it, although sometimes I think you can find those in those signs. But anyway, we also did a lot of extensive collecting on the northern border of Texas with Oklahoma, and that's where you find the appropriately named Red River. We spent time in Arizona focusing on Vitis arizonica. It was at this time when I started realizing a lot more about the geography of Arizona, that there are mountain ranges in southern Arizona that are called the Sky Islands, and I sort of there developed an interest in island biogeography. There's quite a bit, as you'll see here in our herbarium records. We collected several samples that we have dried and preserved in the UC Davis Center for Plant Diversity. There's a lot of morphological variation in these populations, and we're interested in trying to, as best we could, figure out whether there might be genetic differences among these populations as well. The interest in Vitis arizonica was also at the time fueled by some of the plant material that Harold Olnold collected earlier in Mexico, decades earlier, that showed some promise in terms of its breeding potential. I mentioned also earlier the herbarium records. These took us to areas where the environment seems rather inhospitable to most plant life. You'll see the white deposits on the soil below. Those are salt deposits. And you find these in areas of Nevada and Eastern California. The populations in Nevada were also interesting in that they may overlap geographically with Vitis arizonica. And some of the specimens were collected in borderline remote locations, breaking one of Andy's unwritten rules about grape collection, that you don't travel too far from the road to find them, because that's usually where you're going to find grapes in the first place. But Andy humored me on one trip where we drove up into a canyon, finding a rather remote population there that you see in the upper image. We also sampled populations from Lake Mead and found that these populations in Lake Mead were inappropriately called Vitis arizonica. They actually are Vitis gerdiana. And of course, we worked with a lot of different groups in obtaining permission to collect these samples as well. Lake Mead, of course, wanted their samples back, so had to press them and then return them. Around this time, we got an invitation to contribute to the second edition of the Jepson Manual, and this meant a trip to visit many populations of Vitis gerdiana and Vitis californica. We obtained, of course, the appropriate permissions to sample from Death Valley, and several populations were collected from the areas of LA, San Bernardino counties, and then also north and east of San Diego. We have lots of Time spent in the herbarium documenting the samples that are available here in the herbarium in California. And while researching the background of Vitis gerdiana, the initial species description of this, I found relatively thin documentation of the initial description of Vitis gerdiana, aside from Munson described the species and attributed the specimen to the Gerd family, which was from Fallbrook, California. And so our travels by happenstance took us to the eponymously named Gerd Road in Fallbrook, California, and there are still grapevines growing there. Andy was always aware that there are hybrids that are possible between Vitis vinifera and Vitis californica, and so this is also an interest that was that we were interested in exploring as well. The extent to which this occurred, and is there any genetic evidence for this? And so we're answering this question as well. You'll see an example here of plants that were supported to be hybrids of vinifera and californica. And so just to wrap up here on the future of viticulture, of course, works in systematics is always important. It's important to know what you're working with, especially if you're interested in breeding grapes. I wanted to point out that my experience working in Andy's lab was always collaborative and inclusive. You'll see here, for example, Jeff, one of Andy's also former grad students, and Andy collecting soil samples looking for nematodes, and I happened to be along for the trip here looking at the grapevines. And so including folks on these trips was always important. I remember working collaboratively on other people's projects, helping in the vineyards, planting plants. I remember a trip to Lodi where I sliced my finger open cutting Viognier grapes. Not really the best story to tell, but anyway, it happened. I still have all 5, or on each hand, I should say. And it's always great to have technical and scientific assistance from the undergrads, research associates, and the mainstays in the lab like Dan and Samara. So I have to profusely thank both of them for supporting me through grad school as well. Finally, I also want to point out, I mentioned that I'm teaching at a primarily teaching, or I'm working at a primarily teaching institution, and we prepare students for transfer or for career changes. And so I need to point out and put in a plug for the future of viticulture has to include also diversity, equity, and inclusion in the curriculum. I know this is already happening in curriculum, in higher education in particular, but the importance of DEI in the curriculum cannot be understated for an industry like viticulture and enology, which produces a product that can seem kind of exclusive. It's really important to be inclusive so that you're reaching everyone. We like to say in the community colleges, we accept and educate the top 100% of the population. The same is true for viticulture and enology. You want to make sure that you are accessible to the top 100% of the population as well. So I want to thank you all, and thank you especially Andy, your mentorship, your patience with me. It took me a long time to get through grad school. I appreciate the support in my choice of career. I'm very happy where I am and happy and humbled to have been one of your students. Thank you.